When last we spoke about DNA replication, we looked at how DNA is copied on the leading strand of a DNA replication system. Now, we can identify the leading strand because if we look at where the 5' end is at the beginning of the RNA primer, and the 3' end being the 3' end of the last most recent nucleotide added by DNA polymerase 3, we can see that we read 5' to 3' in the same direction that helicase, the enzyme that opens up the double-stranded DNA into single strands, we can see that these two directions are the same. However, if we look at the bottom strand in this situation, so if we extend both strands like this, where the top strand is our leading strand, and compare the direction of helicase movement to the 5' to 3' direction here, we see something interesting. So helicase, if we assume, is opening in this direction, if we look at where the 5' end is of the bottom daughter strand and the 3' end is of the same daughter strand, we can see that this 5' to 3' direction is actually opposite to the direction that helicase is opening. Now, this means that the bottom strand cannot be replicated continuously, but is instead replicated discontinuously, and we'll take a look at why exactly that is. Because DNA polymerase 3 can only add new nucleotides by reacting them with the OH group on carbon 3 of the most downstream sugar molecule in the nucleotide, that means that it can only add nucleotides in a 5' to 3' direction like this, which means if this is the direction of DNA polymerase 3, it's going to run out of DNA to copy at this point, which means it actually needs to jump backward over here and needs to wait for the enzyme that begins DNA replication, RNA primase, to put down another RNA primer in order to give DNA polymerase 3 another OH on carbon 3 as the starting point to replicate the next section of DNA. So because of this fact, because of the fact that DNA polymerase 3 needs to wait until other enzymes do their job, we call this the lagging strand, because it is perpetually behind or perpetually catching up to the leading strand that can be replicated continuously without DNA polymerase needing to finish and then jump backward and start all over again. One other reason why this strand perpetually lags behind the leading strand of DNA replication is that it also needs to wait for helicase. So if DNA polymerase 3 finishes this next fragment of DNA here, it would eventually need to jump forward. Only right now, it can't do that because this section of DNA is already double-stranded. It needs to wait for helicase to open up the helix in order for it to put itself down onto single-stranded DNA and add the daughter strand on top of the newly revealed template strand. For this reason, as well as the reason that it needs to wait for primase, this is why we call this bottom strand the lagging strand, which again we can identify because the direction of replication always moves in the opposite direction as helicase does, meaning that this bottom strand needs to be synthesized in partial fragments of DNA instead of being replicated in one continuous stream. And we call these fragments Okazaki fragments, named after the Japanese geneticist who first discovered that this is how the lagging strand replicates. And if you prefer, in the original Japanese pronunciation, Okazaki fragmento, which again is Okazaki fragments, just pronounced the way that fragments would be pronounced in the Japanese language. Now, this has rather interesting implications to consider about how DNA replication works. Does that mean that every lagging strand of DNA has pieces of RNA that are permanently part of their structure? Well, that answer, as it turns out, is a resounding no, because RNA has a less stable structure than DNA does, because it's actually more reactive. So keeping fragments of RNA in a permanent DNA molecule is actually a very bad idea. So how do we get those RNA primers out? Well, this is where we deal with another DNA polymerase molecule. So in the previous video, we introduced DNA polymerase 3 as the enzyme that polymerizes DNA off of the original primers on both the leading strand and the lagging strand. But here we're going to talk about DNA polymerase 1 because it actually has two roles as far as DNA replication goes. The first of which, we need to actually get rid of the RNA primer, which is why DNA polymerase 1 actually has digestive enzyme activity. For example, if this base on our template strand is an A, that would mean the corresponding base in the RNA nucleotide here would be U. Now, this is a problem because DNA doesn't have uracil, so the first thing that DNA polymerase 1 is going to do is break the bond that holds this uracil nucleotide with the next nucleotide here and remove that RNA nucleotide from the daughter DNA molecule. Now, we know that A pairs with T in DNA, so the next thing that DNA polymerase 1 is going to do is replace that uracil base that we got rid of with the appropriate DNA nucleotide, which in this case would be T. And if this activity applies to every RNA primer, that would mean that DNA polymerase 1 will eventually successfully replace every RNA primer with the corresponding DNA variant. Now, if you look carefully in this diagram, it appears like there is a mistake because it looks like this Okazaki fragment is not actually physically connected with this Okazaki fragment, but that hole right there is not actually a mistake. It's there intentionally to show one of the limitations of DNA polymerase 1. Now, as a polymerase molecule, DNA polymerase 1, like DNA polymerase 3, can only add new nucleotides in, which means that this uracil that it replaced, it can add a thymine in order to replace it, but by the time it gets to the next nucleotide, the enzyme sees, well, we have a DNA base here and a DNA base here, my work is done. And as a consequence of this, it cannot actually connect nucleotides that are already present, meaning this nucleotide, the 3' end of one Okazaki fragment, cannot be connected with the 5' end of the next Okazaki fragment, resulting in this hole between every one of the Okazaki fragments on the lagging strand. And that brings us to the final replication enzyme that is necessary in order to fill in these holes, which is an enzyme that we call DNA ligase. Now, if that name is not immediately familiar, we can derive a verb form from the name of this enzyme, which is the verb to ligate, which is a fancy word that means to join two things that used to be discontinuous together. In other words, DNA ligase's job is to connect the 3' end of one Okazaki fragment to the 5' end of the next Okazaki fragment, and that's exactly what it does. Only it does so in a slightly different mechanism than the way that DNA polymerase 1 and DNA polymerase 3 work. So in DNA polymerase molecules, the way that they work is by first pulling the hydrogen off of the 3' OH and using that to connect to the phosphate. Instead, what DNA ligase does is it changes the activity of the 5' phosphate, making it more reactive and unstable, and therefore making it easier to allow the 3' OH to form a bond with the phosphorus atom here, thereby filling in the hole that used to separate the backbone between the two fragments and turning them into one continuous DNA strand. Now, once this has been done between every single Okazaki fragment on the lagging strand, that is the point at which replication is truly done, and when we have two complete daughter strands, one on the leading strand above in this case, and one on the lagging strand below, as we see here. This concludes the first of our series on DNA replication. To test your knowledge of DNA replication, there is a set of summary questions here in which you need to identify the role of every single one of the enzymes and proteins that is involved in DNA replication and describe why each of these proteins or enzymes is necessary in order to replicate both the leading strand and the lagging strand of DNA.